Before I get going, one, Jenna was asking about what do you actually need for lab on Friday. So, on Friday, what you'll be doing in lab is peer reviewing with another lab group. And by peer reviewing, I mean you should have a really good draft of your unknown presentation in hand. It does not have to be, the script does not have to be complete for it. If you just come in with the script, that's fine. If you just come in with pictures, that's fine. If you come in with PowerPoint, that's good as well. These do not need to be completed, but they should have enough information in them so that when you do a run-through, however formal or informal it is, with another group, they can give you some directional feedback that helps to improve the presentation as is. Okay? At the end of lab on Friday, you need to hand in, just a second, you need to hand in, if I'm not mistaken, it says 2 or 3 things that you and your partner are going to do to make improvements to the presentation based on the feedback. Okay? What I'll be providing you in lab ahead of time will be that checklist so that as you are looking at somebody else's presentation, you can be going through and checking off everything on there to say, what do they have? What's still missing from here? That can provide some direction in what needs to be completed. Okay? Nancy?
So then, do we have nothing due on Thursday?
No? No.
I thought in the duties thing it had the voice fed video due Thursday.
Next week, so next week the final one is due.
Oh,
That'll be due by your lab time. So for this particular class, it's Friday at 10 or Friday at 2.
Wait, so we don't have another assignment?
No, not that I recall. There is one due on Friday. I'll come back to that in a minute. Okay? But I'm just right now exclusively talking about lab. I'm just talking about lab. Cassidy.
So you're going to give us like an objective list of things that we should have done in our project?
Yes, and actually if you're looking for what needs to be in there, this is actually, there is, in the lab manual, there is a page labeled Unknown Video. It has the description of the project. It has a page checklist of everything that you should be making sure that is in there or that has been achieved in some way. Some of them are speaking things. And then it's 3 pages of the rubric that I'm actually using to grade them.
This is where?
It's in the lab manual. It's like page 44 or something like that. So that should all be available to you already. Take a look. Okay? And come as prepared as you and your partner can be to lab. The more prepared you are, the better feedback you can get to help you toward that final product. And again, those are due, the finals are due next week on Friday. I don't need to see these drafts, okay? I don't need to see them. I just want to see what are you going to do to improve on them. So that's one. Is the what? 50. It's a big one. It's a big one. I know. Okay, but I want you guys to keep in mind a couple of things. All of your results, all of your interpretations, including the growth curves, which I'm finishing up I'll finish up by tonight, all of those will have been reviewed by me before this video is completed. So you will have actually already gotten back from me peer review on each of those pieces. Okay? And that's part of my way of making sure the grades on these are better than they have been the past couple of semesters. Fingers crossed. Okay. All right, that's 1. 2, what's due on Friday? So on Friday, you may remember, may not remember, I won't be here. Okay? Yep, everybody's got it marked down, celebrating it as a holiday. Grab the tequila and margarita mix. Across the street, grab the tequila and the margarita mix.
Okay, that works.
All right. So, what do you have due on Friday? A concept map. A concept map on terms that are related to central dogma, replication, transcription, and translation. This is a one-day segue into that conversation because transcription and replication and translation are all targeted by antibiotics, which is what we begin to talk about on Monday. So I'd like you to have just a very broad understanding of what kind of enzymes are involved, so that when we talk about them on Monday, you know where we're talking about, or in the context what we're talking about. This originally was going to be, I've made some modifications from what I said last week. This originally was going to be in your group of 4, hand in 1. I am not making this something that you have to do with your group, but I don't want to review 60 of them. So you need to work with at least one other person. Okay? Has to be at least partner. You may work up to, in total, into a group of 4. Write all the names on it, hand it in, just scan it, and then load it up onto Blackboard. Okay? Paige, I stumbled there. You're looking confused. So where did...
And I think you meant take a picture.
Taking a picture works as well.
Just
If you
remember
wanted to
you
selfie
have
it, that's
to
totally cool.
Do it on like Word.
Oh yes, absolutely. You can do. I'm just saying scanning because people like to hand draw it. But if you want to take a picture of it and then send me a picture, perfect. If you want to do it in PowerPoint or Word, perfect. Just upload it afterwards. There's already a Dropbox created for it. It's in that particular lesson.
That's due Friday night.
That's due Friday night. Originally I had said 2 o'clock, but let's be honest, I'm not going to look at them Friday. So due at 11:59 Friday night. Okay? Okay. We all good with those 2? Let's transition. This was my mood as of last night. So roughly 20 hours ago, this is how my mood went. Epic fail. Why? Because of this. Okay? So, double-edged sword. It's always good to see what people are thinking. It's also really painful at the same time. Because, don't know if you guys know this or not. 1, I'm an anxious person. I actually am on a couple of medications for anxiety. Okay? No, never. Of course. And my wife and I, who both have anxiety, bred, and our children are. Anyway. So, evolution at its finest. So, what I'm going to try to do in the next 10 minutes or so is just kind of summarize my thoughts on this. I'll give some ways that I want to make a couple of changes around here, and then we'll get into the paper. I'm under no assumption that we're going to complete the paper today, so whatever we don't finish talking about, we will start with on Monday as we move into antibiotics. Okay? All right, so my general impression looking at this. 1, I think the prep coming into class is going well. There were some comments on here about, like, I hate group assignments. Fine. You're going to have to work in groups in your life, so in part I say deal with it, but communicate. There are some people in this class that like to get things done really quickly. There are other people who like to take their time and get them done and hand them in at 11:59 the night that it's due. And that's okay. But what I'm asking you to do is to communicate with your partners. Being a married individual, communication is key to any functional relationship. Have to remind myself that daily. Communicate with your partners. So if you feel like if it's an assignment where you have to give feedback, communicate to get it done earlier so that everybody has a chance and doesn't have to stay up until 11:50 to be able to make comments. Granted, what is one assignment we all hate?
Voice threads.
Okay, voice threads are done. Okay, with a caveat. So, I'm not going to require daily voice threads anymore, like we've done for some of these past assignments. But one of the things that I notice is that people want to have some sort of like recap at the end of the week of what are the major points. What did we actually? First exam, am I screwed? No. Simple answer. Is it going to require work? Yeah, I kind of imagine the whole class would require work. One of the things that I do at the end of the semester is I look at the grade as a whole. Just like I ask you guys to look at trends on those graphs instead of single data points, I look at your whole score over the semester. So I look for signs of progress. Are you handing in assignments? Are you visiting my office to come and talk to me? Or are you seeing Julia? Julia and I talk once a week, so I know who's going to see her and who's not. Are your grades on tests improving over time? Those are all different factors that I look at over the course of a semester to make those final grades. So for folks that may be on the borderline, between a CD and a D, or CD and a C, or a B, C, and a C, I take a look at all of that. And I feel like you've put in a good faith effort throughout the semester, and all that's holding you back is one bad grade on an exam. I can overlook that. Okay, those scores there are kind of hard line for a reason, but know that there's wiggle room. Okay. The last thing I suppose I wanted to comment on right now is in terms of like what can you do to grow. I think everybody has a good idea as to what they personally need. This is only good though if you follow through. So what I encourage you guys to do is take that next step. If you feel like you need to do more review problems, set some time aside tonight to do 2 review problems. It'll take you 20 minutes. The length of a Netflix show. Okay? Or not Grey's Anatomy. Not Grey's Anatomy. That's 4, maybe 5 review questions. Let me give you an example. Okay, so which one do I want to share this time? I will share Public Speaking 101. Okay. Given my amazing presence right now in front of the classroom, I was not always this good at public speaking. First year of college, I nearly fainted during a speech in class. First year of graduate school, I went back to my alma mater. My wife and I still laugh at this story today, her more so than me. So I was up giving a talk, high school students in the crowd, and I made some really bad joke, and nobody laughed. Nobody. It was just like blank faces. Nowadays, I just say whatever, but back then, it freaked me out. I started holding on to the podium, and I was like 2 minutes from finishing, and all of a sudden, everything started blacking. And I was at the podium holding on and started stumbling on my words and started like shaking like this and like falling down. My wife had to come like grab a chair to set me down from fainting. And that was horrific because they just stopped the speech there and were like, okay, thanks. And I wasn't invited back for 5 years afterwards. So why do I bring that up? Because I had a huge fear of public speaking, okay? What I found worked for me was, it took me a long time to figure this out, but what worked for me is like, I realized that it's not me, or I'm not trying to present myself. I still can't do that. What I can present is data. What I can present is class material. And it took a lot of practice for me to realize that's what worked. So, you know what you need to do. You now have to take the time to figure out how are you going to make that work. Okay, that's what I want you to be able to do. So and next time you're going, I need to do review questions. Just imagine me passing out in the middle of class. All right. Anything to add, or any other comments?
Do we get the extra credit?
Yeah. So I looked at the number. No, that's a great question. I forgot to mention this morning. 53 is roughly like, it's somewhere around 3/4 to 5/6 of the class, 2.5 points. That's how I measured it out to. Yep, every single person in the class gets 2.5 points extra credit. I will post it by the end of the weekend. There will be an extra credit tab now. Oh, by the way, there's more coming up in the next week as well. It's glass though. It's glass on the floor and you have to walk across with your bare feet. Who would do that for extra points? That's awesome. Fools though, for extra credit, that might be worth it. Anyway. right. Let's talk about this paper. So, Mancini, you just looked at the figures.
Okay, if you're gonna ask about the figures, I'm only gonna look at the figures.
Fair enough. Those are probably the most important points to take away. I will fully agree with you on that. What we're going to do today, I'm not going to ask who liked this or who didn't, because the answer is going to be like, please don't let us ever have to do this again. I can't make promises, especially that one. Because as much as it sucks to read these sometimes, like some really cruddy medicine, it's good for you. This is a good thing for you to do because this basically is forcing you to apply everything that we've talked about in the past 2 weeks. We've talked about growth control, and we've talked about ways of measuring growth. This paper hits on both of those, okay? So what we're going to do today is try to walk out of here at least with an understanding of half of this paper, and we'll finish up the rest of it next week on Monday. The way I want to do this is kind of like basically a jigsaw. So I've given 9 different prompts. You and your partners are going to have to answer that prompt to the best of your ability. I'm going to give you about 5 to 10 minutes to do so. And then after you've done it, we're going to report out. Okay? And the way that we're going to do this is just whoever is the person writing down the notes will report them out. And then we'll talk about it. And what I want you to do is, if you are the person recording the answers, modify. Okay? If there's modifications that need to be made, make them right on there. Because what I'm going to do at the end is I'm going to collect all these, I'm going to scan them all, and I'm going to post them up on Blackboard. Basically a study guide of this paper. Okay? All right, so, and we're going to start to talk about then what makes a good response better from a great response or an excellent response here. When it comes to a scientific paper like this where you have it in hand, it comes down to the details. Okay? It comes down to being detailed oriented in a paper like this. So each group has a different one. I think that, for example, the first one is something about, well, actually, so what is number 1? Who has number 1 in the top right corner of their page? You guys do, right?
Yeah.
Okay. So once we go through, you guys will answer that question for us. Did you already write down an answer? Good, I'm giving you number 3 as well. But blame Paige for the rattiness of this page. She moisturized. I said blame Paige for the rattiness of this page, though. She moisturized. All right, so we clear with what we need to do? For those that have one that says diagram, it means draw a picture of basically the experiment. How did they run the experiment? I'll be walking around to help out. There you go.
Describe the chemical results. Figure 1, make sure you describe the measurements of X, Y,
Yeah.
So you guys started the script for
Versus after.
There's also like
I named that graph, right?
I mean, it wasn't wrong,
Well, was it right?
but I mean, you could have added more details.
And then interpret the results.
I just think,
I guess.
I think the name of our
I mean,
describing
definitely,
coded results
obviously,
for
after
the
we watch either so before that, it decreases. Yeah, there's not really a system.
okay.
This is just, there's.
So it's bacteria,
I think the only difference between A and B, figure 1 was one's gram positive bacteria and one's gram negative bacteria.
Was it, or was it the,
Or, no,
I think it was
it's
the
the difference.
temperature.
I think that's both soaps.
Yeah, it has plain soap and it has
And the difference between figure 1 and 2 is the temperature.
All right, so I am not grading anything here. This is just based on, this is just how well do you understand it. Okay, the goal is to try to take our understanding, and if we need to make modifications to it to increase or to enhance that understanding a little bit. So, you guys get to start out, right, Jenna?
Yep.
So you guys had, what is the research question? What is the importance of the question? And why was the FDA considering a ban on triclosan enhanced soap?
So the question is antibacterial soap containing triclosan more effective than plain soap and water? This is important because about $1 billion are spent annually on making the antibacterial soap, and FDA is considering banning it because they has adverse effects like allergies, antibiotic resistance, endocrine disruption, and some other weird stuff.
Yep. So basically the question comes down to do the drawbacks outweigh the benefits or vice versa, right? That's really what we're kind of getting at here. And I want to, will you read your research question again real quick? I want to make one modification to it.
Is antibacterial soap containing triclosan more effective than plain soap and water?
Okay. More effective than plain soap and water at what? Killing bacteria, or let's think of this another way as well. I think that's fine. Let's just make a slight modification to that now. When we wash hands, how do we categorize that? Is that considered to be a disinfection? It's a degerming event. And what is the goal of degerming? What's that?
Get rid of germs.
To get rid of germs. In our definition, at least at the way that we used it, it wasn't about killing per se. It was about mechanical removal in some way. And so I think, Jenna, I'm not trying to say that question isn't correct, but I think one of the things that we have to consider here is think of what the use of triclosan in hand soap was, or at least how we understand it. We thought of it as an antibacterial, something to stop growth or to kill bacteria. But in essence, how long is the triclosan on our hands at any given point? How long should we wash our hands?
30 seconds.
20, 30 seconds. I will bet you if you pull, not this floor, I bet this floor is pretty darn good. But anywhere else on campus, if you were to just go ask 20 people at random, how long do you wash your hands for? Do you say greater than, less than, or around 20 seconds for most people? I would say way less, right? And so remember what we talked about. Remember what we talked about on Friday. We said that there are two important factors to consider how effective an antimicrobial is. One is the concentration. What's the other thing that's important? So the ability of an organism to survive a chemical treatment is dependent on concentration of that chemical. Zach?
The time of exposure.
Time of exposure. And time of exposure here is 20 seconds. Is that enough to kill a microorganism? The answer is probably, well actually we know the answer based on some data they show later on, but the answer is probably no. And so that's something we have to keep in mind here, right? If we're considering the use of triclosan to reduce bacteria on hands, how long would we have to allow it to incubate? More than 20 seconds. Okay? That's something for us to think about. But I don't think that at all really affects the question at all. And just something I wanted to add on to this. Okay? Anything else to add to what's been said? One of the other additional issues, Jenna, did you mention antibiotic resistance as potential? Yeah, and that is another key issue here. And we have found there are examples where bacteria have gained resistance to triclosan as well. So something to keep in mind. Okay. 2. Who had the second one? Okay, so how did the authors decide on the experimental design and what led them to choose the time, temp, and percentage that they did? And then we'll go to the last one. Yeah.
And then that was just the time considered was where it went up and washing your hands. And then instead of, you say triclosan?
Triclosan.
Triclosan of 0.3%, this is a maximum determined by law.
Okay. So basically all these are based off of standards, right? There are 4 or 5 major entities that have these. A typical hand washing should be 20 seconds and enough time to sing happy birthday to yourself twice, and then that concentration is the maximal amount allowed. The 40 degrees Celsius, where does that come from? I think it says something about it in the next paragraph, that there's another question. I apologize, but the triclosan's a bisphenol. So it falls with the bisphenols, and the role of those, again, they disrupt membranes, but the soap itself is a detergent or a surfactant. We talked about how those are really good at degerming different surfaces. All right. Who had this one? Diagram the experiment. You want to bring it up? Do you mind explaining it, too? Sorry, I forced you to do more than you expected.
Yeah, well, those are just the cells in the tubes, the cells with the plain soap, and the cells with the triclosan, and then they're incubated at the 22 degrees Celsius, and then they are serial. That's like a serial dilution, and they are plated and counted.
Yeah, exactly. So in essence, what they are doing here is taking those cells. This is this first experiment, 1 and 2, not on hands, in a test tube. Incubate. They add something in the middle there to inactivate the chemicals that are in to inactivate the triclosan, for example. Serial dilute and plate count. And what is our measurement here? When we do a serial dilution and plate count, anybody remember, not measurement, but the equation that we use? CFU divided by what? V, which is the volume that you plate, and D, the dilution. So the numbers that they're reporting are the calculations from those plates that they're looking at. Perfect. Perfect. All right. How does figure 2 differ from figure 1? What's the major difference between 2 and 1?
Temperature.
Temperature. So the figure 1 was done at 22. Figure 2 is done at 40. And then everybody pick up on the difference between A and B in both of those figures. What's the gram positive and gram negative. So 1 represents just a classical set of gram positives that have been used for these types of experiments. The other one represents the gram negatives. And then Kevin had asked, what do those numbers actually mean in those graphs there? Not the data, but rather the numbers here. These are specific strains of bacteria that they're using. So it's a specific strain. Oh, gosh. My mistake. Thanks, Paige. Those are specific strains. So like this L monocytogeny 7644, it's just a designation so that if a lab here is doing the same experiment as a lab in California, they can use the same exact bacteria. That's the design behind it. All right. I think the fourth question is analyze this graph. Am I not mistaken? Who's got that one? All right, go ahead.
So, figure 1, like we said, it's at 22 degrees Celsius for 20 seconds. A is gram-positive, B is gram-negative, and it's shown to be bacterial population. That's the white bar is the initial population, and then the lighter gray bar is the population after plain soap for 20 seconds, and then the darker bar is population after antibacterial soap for 20 seconds.
Okay, great.
Not much difference between antibacterial and plain soap.
Okay.
As phylogenous.
Yeah, and we can come back to that one. So a couple of things to point out. The first, obviously, is you've got the x and y axes labeled correctly. You've got the bars labeled correctly. What is our one? But how do you actually read this graph here? The biggest thing. What is a log of 0 equal to? 1. So another way to look at this, if you ever see a log base table like this, just put 10 below it so that that number is the exponent. So 10 to the 0 is 1. 10 to the 1 is 10. 10 to the 2, 100, so on and so forth. So in other words, 10 to the 6 there, that's 1 million cells they're counting. 1 million cells that they start with per milliliter when they add the soap. It drops down to 10 to the 5th or 100,000. In other words, if you were to make this a percentage, how effective is washing your hands at removing bacteria? Yeah, it's 90% effective. 90% effective. Why do I bring this up? Mostly to make a point that in the world of bacteria, we consider tenfold changes to be significant typically. Ones that are like twofold, going from 200,000 down to 100,000 cells, doesn't make much of a difference. Case in point, if you ever look at, for example, Lysol disinfectant wipes or toilet bowl cleaner, how do they report the effectiveness of those chemicals? 99.9 or 99.99. In other words, for every 10,000 cells you have, all but one of them end up dying. That's effective. Okay, that's effective. But saying that there's a 50% decrease in bacterial numbers, that's not effective. That really isn't big when we consider microbial numbers.
I thought they just did that so that they could, like, if you get sick after you use it, just be like, gotcha.
Well, I'm sure there's a little bit of that with it. I'm sure that it's a little bit of that. But I guess I always quantify it back to, like, well, what does this actually mean on a grand scale, right? Now, granted, at the same time, if you start with 1 million cells and 99.99 decrease, that goes, let's see, 1, 2, 3. You've still got 100 cells around. Guess what? Guess what the infectious dose for E. coli 0157H7 is? Yeah, it's a single cell. Is enough to cause illness in some individuals. So, yeah, just keep that in mind next time you start Lysoling the crap out of everything like my mother-in-law does. It is annoying as hell. Back to this, so 2 points we can make out of this one. 1, does hand washing have an effect? Yeah, there's a tenfold decrease. Not only that, but if we take a look at the error bars, there's no overlap. And that at least would suggest to us that there is significant decrease. Does the triclosan have an effect? Not at 20 seconds. And the reason why I want to make that point, this will be my last point for today. Somebody in their response, again, I don't remember which class, said a comment like, the antibacterial soap had no effect compared to the regular soap, which makes no sense because it's antibacterial soap. And the point I want to make is your interpretation should not guide your analysis. It should be the other way around. Analyzing this graph, and results don't lie. Okay? Results tell us what's really happening. Our interpretation is what can be faulty here. So if you use the results to guide your interpretation, rather than taking what you think is going to happen and try to make that explain the results, that's the way we should be approaching science. Right? That's the way we should be approaching this. we'll finish this up on Monday. So just kind of put this aside. I will take the sheets so that we don't lose them though. Alright? Otherwise, enjoy your Friday without me. Don't cry too hard. Don't cry for me. Argentina.
Yeah, I'll
Next week, yeah, just next week, we're working on it.